Okay, good afternoon and welcome back to this session. So it's again a pleasure to introduce Professor Deepa Agashe from the National Center for Biological Sciences right next door. So Deepa did her PhD from University of Texas at Austin, and after postdoctoral work at Harvard University, joined NCBS some years ago. And she has been doing really fascinating work across a lot of different disciplines that include molecular evolution, codon usage bias during protein synthesis, and behavior of beetles. So behavior of beetles is what she's going to talk about here. So over to you.
Thank you, Supratim. I hope you can hear me in the back. It's a pleasure to be here. I think the crowd is quite diverse, so please feel free to stop me if something is not clear. I can explain in between, because otherwise the rest of the story will not make sense. So as Supratim said, I'm going to tell you about decision making in an insect. And this is an insect, or a much prettier picture of it than what it really looks like, drawn by a very good artist. So this is a pest species that's found everywhere, and it's a very handy and useful model system that we use in the lab to do experiments. I'll tell you more about it later. But let me start off by making just a couple broad points. As you probably are aware, animals are faced with lots of choices about what to eat, where to sleep, where to take shelter, and so on and so forth, at every point in their lives, and we are no exception. So I suppose you all had lunch a long time ago, so this is not going to cause any hunger pangs. But imagine a poor insect in a milieu like this, right? There are about 30 whatever species of possible host plants that it has available, and it has to figure out which is toxic, which is not toxic, even amongst the ones that are not toxic, which ones have the right nutrient content to be able to sustain, to maintain high fitness, how to optimize fitness over long term, and so on. And that's the core of what we're thinking about. And people have been asking for a long time, what factors contribute to decision making in animals? And insects have been studied a lot for this. I'll just give you one example to start off with. We think that competition is really important to decide what an insect is going to choose. And this is important because if there's a lot of competition for your favorite, best, optimal food, then maybe it's better to explore something that is not so optimal, right? So here's an experimental study from many years ago, where Dan took flies, just normal Drosophila in the lab, and gave them either high competition or low competition by having them at two different densities, right? And then he gave them a choice of normal media. This is just the fly food that you might have seen in labs, or media which is laced with toxic cadmium chloride, right? So the typical fly will avoid laying eggs and eating media which has cadmium chloride. But what you see here is what happens after 4 generations of evolution and selection with these choices offered every generation. You see that flies which have higher competition, which is the squares, are much more willing to go and explore and eat cadmium chloride, which is toxic, compared to flies which are not experiencing high competition, right? So very, very rapidly, behavioral choices can evolve in response to competition as the main factor in this case. So many such factors have been studied. People are interested in asking how decisions are made. And what we saw sometimes is that sometimes animals will make different choices than what seems intuitive. So I showed you before that flies seem to choose suboptimal resources when they are faced with high competition for the good resource. But we saw that in flower beetles, which is the same model system I'll speak about later as well, if you give them a choice of wheat flour, which is optimal for them, and corn flour, which is really bad for them, and you vary the population density, we expected to see something like what Dan had found earlier. Instead, what we see is that the amount of corn being used is reducing as the populations get more and more crowded. So they seem to be avoiding the new resource at high densities. And the same thing is also observed if you look at how many eggs the females are laying in these 2 resource patches, corn and wheat. So at low density, females are laying about 80% of their eggs in corn. But at high density, that reduces to about 60, 70%, right? So it's going in the opposite direction to what you might think intuitively should be going on. And we have spent a lot of time after this asking, why are animals making these kinds of apparently suboptimal choices? So we thought, okay, maybe what we're doing is we're not measuring things properly, right? We're not measuring what is actually relevant for fitness or for this decision making. So one of the things we found out, this is work done by my former PhD student, Vrinda, is that if you expose a larva of this flower beetle, so the flower beetle goes through egg, larvae, pupae, and adult stages. And if you expose larvae to 4 different environments, 4 different resources, 3 of which are bad and 1 is good, depending on what it has recently tasted, it is much more interested in exploring that and eating it later. And maybe if any of you have observed kids, you might see the same thing, right? So if you expose children at an early stage, this is scientific studies, by the way, that have been, not just anecdotal, right? Expose them to different kinds of foods at an early stage, they're much more likely to be non-fussy eaters later on as well. So it's sort of the same thing we're seeing. If you offer larvae a choice of finger millet and wheat, but before that you expose them to finger millet, they're much more likely to choose finger millet later on. But there are limits to what they will do, right? So if you give them a terrible resource, even if it is offered a taste earlier, it will not choose it. So there are limits to what it will decide it will not do. But what's important is that because larvae can now go and colonize new environments, if they have experienced some new resource before, they're much more likely to be able to colonize these new environments more successfully potentially, as long as it's not reducing fitness too much. So it's important then to be able to ask why or understand why animals make the choices they make. The second kind of study we did a few years ago was to ask whether female context matters, right? So if we take females and we give them a choice of wheat and finger millet, like in this petri dish here, we can then go and count how many eggs the female is laying here versus here and ask what is that choice that this female is making. What we found is that depending on the context from which the females came, that choice is very, very different. So I'm showing you here 2 important aspects of this oviposition behavior. There's fecundity and preference for finger millet, which is on the y-axis here. If you take females which are young and come from a not crowded environment, this is gray bars here, you see that they have high fecundity, so they have lots of eggs, and those eggs are being allocated sort of randomly, right? So half the eggs roughly are in finger millet and half are in the wheat patch, which again seems a bit counterintuitive. Why would they not lay all their eggs in wheat? But if you take females which are young from high density, so crowded environments, they really avoid laying eggs in wheat, in finger millet, sorry, and they have much lower fecundity. And similarly, with a third different context, you get different results as well, right? So basically, depending on what the female has experienced in her early life, she's making very different decisions about where her offspring are going to develop and what their fitness might be. So to understand this a bit better, we said, okay, let's manipulate what's happening with the females and the eggs. So let's create a fitness landscape of sorts. So we manipulate egg allocation. So we take females, let them lay eggs in wheat, and we take all the eggs that a single female has laid and either put them in only wheat or only finger millet, or distribute them equally or quarters and so on, right? So now what we're doing is we're taking the choice of egg allocation away from the female, but we retain her natural fecundity. And we do this for many females, right? And then we ask what happens to the fitness of these offspring that develop in the patch that we place them in. So we ask how many of the offspring become adults, how many total offspring are there, how fast do they develop, and so on and so forth, and what proportion of them survive. When we do this, we can then construct a fitness landscape like this, where as a function of fecundity and egg allocation, that is what proportion of eggs are in finger millet, we can construct a fitness graph like this, where we can say yellow is high fitness, the blue and purple is low fitness, and we can ask at what parameter space are we getting highest fitness values, right? And then does that change based on context? So we can take females that come from different contexts, low density, high density, less crowding, more crowding, old, young, and so on. And then we can ask what happens to them when we manipulated everything, right? When we don't give them a choice. So what do we see? Here's what the data look like. Again, it's the same kind of plot I showed you before, fecundity on the x-axis. On the y-axis is percent egg allocation in finger millet. Every dot here is a single female and how many eggs she has laid and where she placed them, okay? Where we placed them, right? So her choice is no longer there. And then we ask, what is the survival? So the yellow, you can see here that in this left corner is where survival is maximized, which means that at low fecundity, high allocation to finger millet, maximum survival is observed. In contrast, if you look at development rate of offspring, so where they develop fastest, this space seems to be the highest fitness. If you look at total offspring, this corner is the highest fitness, right? So already what you can see is depending on what we choose as our fitness metric, we would get very different optimality criteria. So here's what that plot looks in if we smooth it out and create a fitness landscape from that, which is the background here. And you see what I've just showed you. This left corner is the optimum here. this corner is the optimum here, and this corner is the optimum here, right? Depending on the different fitness values. And what you see, which is most interesting, is that if we take our old data, which I showed you earlier from these females laying eggs, when they had a choice, right? What were they doing? The old HD females seem to be closer to the left corner. The young LD females, on the other hand, are closer to this right corner, right? So it suggests that depending on the female context, they are optimizing different fitness parameters for their offspring. And you can see this in this plot as well. If you look at how many total offspring a female can produce versus how fast those offspring develop. So in a given fixed amount of time, do all the offspring make it to adulthood or do they not, right? The gray points are the young, low-density females that I showed you earlier. You see that they have high fecundity, but their offspring don't develop very fast, correct? And this is partly because they allocate their eggs randomly to finger millet and wheat. So on the whole, they don't develop as well as the others do. But the other two contexts, females from the blue and the yellow context, which are older females or younger females in a high density context, they are allocating or optimizing offspring development rate and not total number of offspring, right? So this suggested that if we want to understand decision making, we need to be able to understand what are the appropriate fitness parameters. Now, more recently, we wanted to understand how this or why this kind of trade-off is occurring and what's giving rise to this decision making. How do they make sure that these offspring are, why are these offspring slower developing versus these ones are faster developing? So we started doing some simple biochemical assays to ask what's the content of these eggs that females are laying when they come from this context versus that context. That's what I'm showing you here. So we did a bunch of assays for lipids, carbohydrates, proteins, and so on. And we find that lipids are the one parameter which just seem to change across different eggs. So if you count how many eggs are there and then ask what's the total content, divide by the number of eggs, you get how much lipid is present in each egg of a female. Again, every dot here is data, average data for a single female. You find that as females are older and coming from higher density, they're provisioning their eggs with more lipid, right? And we don't see these differences for protein or other things that we've measured. So what this suggests then is the reason why, one more thing I should mention before that, is that the reason this kind of trade-off is seen is that as fecundity increases, the amount of lipid per egg is reducing. This is true across all the contexts, right? So what this means is that when females are laying more eggs, they're not able to put the same amount of nutrient in all the eggs. They put less nutrient in all those eggs. And that potentially explains why those eggs are slower developing, right? So we think we have some handle on the mechanistic basis of what's going on and what drives these differences across different female contexts. So to summarize this part, what I've shown you is that behavioral choices can shape the use of new habitats. There are strong effects of prior experience, and context-dependent behaviors can allow optimization for different fitness components. So next we wanted to ask, what is the impact of all of this, right? So in the short term, I've shown you that it might matter for the next generation. Whatever decisions the female is making in this generation can change what the next generation is doing or happens to it. We wanted to ask about evolutionary time. Over evolutionary dynamics, evolutionary time scales, does context-dependent choice of the founder females matter at all? One way that they could matter is if there are female maternal effects, right? So the female is coming from context A versus B. She makes different decisions, and there's these percolating ecological effects through population size, density, those kinds of things. Another possibility is that if the female is putting something in the eggs that is changing how they are going to develop or their behavior in the next generations, that could percolate. Or we may see nothing, and this whole experiment falls flat, right? So of course, it didn't fall flat, which is why I'm telling you about it. So we manipulated age and density in a full factorial experiment. So we have either young females or old females, and they come from either a low density or a high density context, and we can manipulate this nicely in the lab. Yes. So we take these females and we allow them to establish populations in the lab, which we then evolve in this patchy environment, right? So they get a patch of wheat, a patch of finger millet, and then they're allowed to lay eggs for 1 week, and they get transferred. I'll tell you about the details of this. But the goal here is to ask, do population dynamics change depending on which of these kinds of females are the foundresses? Does the oviposition choice behavior change of these females over evolutionary time? And for how long do we see the maternal effects percolating, if there are any, right? So we take females that are isolated and allowed to age for 2 weeks or age for 6 weeks. So that is our young females, initially young, and then they age for longer or shorter in isolation, right? So they're not in a crowded environment. Or we take females from stocks where we know exactly how long the females have been growing up after eclosion, either again 2 weeks or 6 weeks, right? So that's the context from which we derive our females. And then we take these females, create 6 replicate populations. Remember that this context that we're manipulating is only there for the founders, right? We change the founder females, they go in, they lay eggs. After that, we don't manipulate anything else. What we do is we take 100 adults from each context, we allow them to lay eggs and colonize this kind of habitat where half of it is finger millet, half is wheat. There are no barriers, so they can freely move across and do whatever they want. Larvae can move, adults can move. We census, we allow the females to lay eggs for a week. We remove all the individuals. We census the population after 6 weeks. So this allows time for individuals to grow up, become adults. We count how many adults, pupae, larvae do we see. And we take 100 adults out of whatever we have and put them back in, right, and start the next generation. So in this process, what we're doing is we're selecting for individuals that can grow reasonably rapidly, right? Within 6 weeks, if they are adults, then they have a chance to go to the next generation. Otherwise, if they're larvae, then they're dead and they're in the freezer at the -80 at the moment, correct? So this is what the flower looks like at the end of the 6-week cycle. You can see that it's all mixed up, right? There are no distinct patches that you can easily tell apart. They're sort of there, but not very clearly. So then our goal is to ask what's happening to their resource use, what's happening to their fitness, their oviposition choices, population dynamics, and so on. So here's an example of what the population dynamics look like. So on the y-axis is total number of adults. This is from all the census data. The experiment went on for 16 generations, so we did 16 such transfers. Every transfer we have whatever number of adults, and you can see it goes up and down. This is entirely expected because the species has negative density dependence. We expect that that's going to kick in. Anytime there's a boom in the population, typically next generation there'll be a bust, right? So this keeps happening. The thick lines show you the median trajectory for all the replicates, right, for a given environment. The dotted line shows you median population size, and you can already see that the young LD has a higher median compared to the old LD. So there's already some differences that we see, but the cycling is not particularly different. And if we do several kinds of time series analysis, we don't see any differences in the cycling. What was interesting, and this happens in experiments once in a while, is that we had an unintended period of parasite that sort of swept through our whole populations, right? So this is the part that's highlighted in gray. This is when we started noticing larvae were dying, they were shriveled up and ugly and black and just disgusting. But anyway, so this is parasite, something is known about it, not much is known about it. So I don't know exactly what parasite it is, it's a microsporidian. It kills larvae, that much we know, don't know much about, much else. Okay, so then what happens to these other contexts? So we have young HT and old HT as well. And you see again that there's the cyclical dynamics, and at least eyeballing it, nothing looks particularly different. If we ask what's the population size, the harmonic mean population size, given the fluctuations, we do find that founder age and density context matters, right? So the young LD females that founded these populations across 16 generations, their populations tend to be larger. And it's not a tiny difference. It is a bit of meaningful difference, I think, because it's about 150, 160 versus 70 or 80 or so, right, for the other populations. So potentially there are consequences of these founder effects on population size in the long run. We also find that during this parasite infection, if we ask what happens to juvenile development rate, it reduces in general. So the higher the parasite infection, estimated using the number of dead larvae that we find, the lower the juvenile development rate. So it takes longer for larvae to develop when there's a parasite around. And that the strength of this relationship, this deleterious effect of the parasite infection, is also dependent on the context. So perhaps what your, I don't know, 15 generation grandma did in terms of her life and her early experiences somehow seems to matter for parasite infection 10 generations later. This is super surprising to me. I don't understand it fully well, but it's really, really fascinating. All right, so then we wanted to also measure oviposition choices. So we place the female, as I showed you earlier, in between this choice plate. And we have a trick here where to separate the eggs, we cut the plate in half and then measure the two parts separately and count the eggs in each batch. And then I'm going to show you this plot, which is going to be a bit complicated, but it's similar to what I've shown you before. So it's total finger on the x-axis, preference for finger millet on the y-axis. And the gray line is the kinds of data I showed you before, right? So we take females, they go through their context, they get prepped, they age, whatever, and then we put them in isolation and we measure how many eggs they lay and where they lay their eggs, right? But in our populations, remember, there are 100 individuals together in that population, about 50 of which are going to be females. So they are exposed to that group setting for a week. And we also wanted to ask, does that change their behavior in this context? So we take females in such a group setting, which is the green dot here, and we ask what they do. And you can see that within that 1 week of exposure to a group setting, their preference and fecundity dramatically changes, right? So it goes from this gray to green. I'm showing you here data for this young HD to start with, just because it's simpler. And then what's really also interesting is going from this 0 generation, what's the foundress doing in the population, to 15 generation females, there's again a big change, right? But it's still smaller compared to that initial immediate jump in the oviposition choices. So decisions are very, very malleable, changing within a week, as well as across 15 generations of evolution. What happens in the other contexts? This is where things look a bit crazy. So I want you to focus again on the gray points first. The gray points are the starting single females. You see one here, one here, one here, one here. So there's a lot of divergence in what they're doing initially. This you've seen before in the previous data I showed you. The green dots are informative. They say, okay, even after the foundresses have been in the habitat for 1 week, they are still all over the place, right? That's the green ones. And the pink ones, interestingly though, all come together. So by the time we have 15 generations of evolution, everybody's behaving in a convergent manner, right? They sort of have high fecundity, but intermediate preference for finger millet. Or you might say it's no preference for finger millet, given how close it is to 50-50, right? But that's a very big shift from where they all started off, right? So initially there's divergence across populations and contexts, but then there's convergence over time, maybe indicating that the effect of maternal founder effects is slowly reducing over time. So that decision-making ability and choices that they're making are important for some time, but after a while, maybe not. And you see this also in this analysis. So if we take all of our data together and we ask, how much does fecundity or preference get affected across all these different generations? We measured these 2 different traits for females, and we ask age density or age by density interaction for the founders. What is the effect size of these, right? So it's just simple general linear models that we use here. You see that, and the asterisks indicate cases where the effect is significant. You see that the effect is tending to be more significant in the early generations, and the effect size also reduces later on, right? So it supports the idea that the maternal effects that we see, the founder effects that we see early on, are going to disappear over time. But I'm still surprised that they play that big of a role even after 10, 15 generations at all. So to summarize this part then, what I've shown you is that founder female context initially drives divergent oviposition behaviors, very, very different from each other. And these effects reduce over time, which allows for populations to converge ultimately. And we know that these founder contexts affect population size, and they also influence parasite resistance to some extent, right? So there's a lot more work to be done here. We are asking whether the kinds of lipid and protein provisioning that I showed you for the initial founder context, how has that changed? Has that changed at all during this course of evolution? Or has the trajectory of that provisioning also changed across contexts, across time? What has happened to these behaviors in conjunction with the diet? So I haven't told you anything about what these beetles have eaten, right? They had a choice of finger millet and wheat in front of them. So we're going to do stable isotope analyses to ask what these beetles have eaten and how that diet itself has changed over evolutionary time. And the hope is that we can then correlate what the females were doing with what their offspring were in terms of their fitness, and what the choices of the females were. Do they reflect the diet in any way at all? And does that change how they provision their eggs in any way? So this is where we're at the moment. I'll end by thanking everybody who's been really instrumental for this work. So the first part of the work I showed you was done by Vrinda, former PhD student, and Gaurav was a JRF in the lab. Shubha is a current PhD student who's done all the later evolution experiments that I showed you, and I think she's going to find really, really interesting things once she does all the other assays that are remaining. Dhruva was again a GRF in the lab who helped Shubha with her experiments. I thank my entire lab group. This is a bit of an old picture, but most of the people are there, who are a really fun bunch to work with and make it easy and much more interesting to work. NCBS for funding and support. And if you'd like to know more about what we do, please visit our lab website. I'm happy to take questions. I don't think we have enough data to really get to those level of details, but we haven't tried it. So it's possible that we can extract that kind of information. Yeah, we haven't tried it. Yeah.
Question here. So these adaptations that you sort of shown, this is at the level of phenotypes, right?
Yes.
So have you tried to figure out that whether this already has done something to the genotype and that is getting reflected in the phenotype?
We haven't tested genetically at all, no. It's not trivial to do for these beetles because the genome is quite large and the populations are, yeah, anyway, so it's not trivial to do that, yeah. Yeah, I mean, people have certainly seen that. What I don't expect based on other work that we've done in the lab, so if we evolve populations for even 50 to 100 generations, we see at the moment very few new mutations fixing in the populations. So I think in these kinds of studies, typically it's standing genetic variation that's driving adaptation much more. So my expectation in general would be that's what's happening here. So we're starting our populations with a fairly outbred line of beetles. So we collected beetles from different parts of India, brought them together, made an outbred line, and then set up the experiments. So my guess would be that it's initially existing variation which is being sorted in this process, and I would expect very few new mutations to play a role, unlike what we see with microbes, for example.
So if there has not been much of it, genetic changes, so which machineries must have?
No, so I'm not saying there will not have been genetic changes. I'm saying new mutations are probably not driving what's going on. It's existing variation which is being sorted out, right? So for example, I'd expect that the variance in trait values is probably reducing over time. We haven't tested that, but we'll test that. And the other problem in some sense in addressing what you're trying to ask is that I think these kinds of phenotypes that we're measuring are extremely plastic. And you see that also in the data that I've shown you, right? So I would hesitate to bet that there's a lot of, there are a few high effect alleles or mutations that are, or genetic variants that are important for driving these kind of behavioral changes, right? So I don't know exactly what has happened, but my guess is there is still a huge plasticity component which is still playing a role, but I'm certain there is evolutionary change that has also happened, and at the genetic level.
Thank you.
Hi. So when you start with like a young female in a situation where there is less density, and then you show that there are cycles of the population, so the next generation, the probability density is going to be high. So do these new females actually behave similar to young females in a high density, or do they sort of, because of, I don't know, epigenetics or whatever, that they actually stick with their mother's behaviors more closely?
Yeah, good question. So we tried to ask that. We're not seeing any obvious clear patterns. So there's also a lot of variation across the different replicates that we have. So overall, in terms of what we see for the first generation female behaviors, there isn't a clear effect of what their mothers were doing. Yeah.
What about like age? If you just switch age as the thing while you keep sort of the pressure of density the same.
Yeah.
Oh,
so again, the founders behave very, very differently depending on their age for a given density context. What their offspring do, I think, also does not show a clear mother's age effect. It's a bit mixed.
And if you pick up individuals which are going through these experiments in the middle of, say, and I don't know if this experiment has been done, but, and you sort of make them as new founders. Is that interesting at all, or
what might you expect to see there?
So sort of like if they get a chance to found a population while they have already gone through this history of stress, does that impact the future or not?
So I should say that they're actually not experiencing too much stress per se in this
Okay,
environment.
yeah,
I mean,
yeah.
they have their, at least a bit of their nice resource which they like. And the population densities almost never go insanely high.
Right, okay.
Because there are these negative density dependent mechanisms that kick in reasonably fast. So I don't think they're really experiencing stress so much, but of course the density differences do matter to them. So we've asked whether whenever we've measured in between what the females at that point are doing in terms of their oviposition and so on, is it correlated with the immediate population density that they experienced when they were growing up, right? And we do see some patterns there that make sense given what we've seen before.
I see, okay.
So there the founder effect doesn't matter so much for what these females are doing,
Right, right.
but their immediate density does matter. And you'd expect that. That's kind of what you're asking, right? So,
And just one final, yeah, just one final question. Just the 6 weeks that you chose in the first experiment, was that a non-trivial choice, or
Yeah,
because
it
it
was-
reflects similarly to their life cycle?
yeah, yeah. So after 6 weeks, their fecundity drops a lot, or around 6 weeks already.
Just to understand your experiment better, did you isolate the female in some other petri dish or something to lay eggs, like while she lay
Yes.
eggs? Then won't she behave differently when she is in the community surrounded by other individuals? And
Yeah, but we can't do anything about that, right? Because in the community, we can't ascribe the number of eggs that we see to a particular female.
But
Then
you
we just have
can-
group level effects that we would see.
yeah, you can say that, okay, there are 10 females and there are 200 eggs, then maybe it's 20 per female.
We could, yeah, so we've tried doing that, but it turns out it's not very accurate. And the other problem is that there is a lot of variability in a given box in how many eggs each female will lay when we pull her out and do this work. So I would hesitate to make those. And then my student would completely abandon her PhD and leave if I asked her to count all the eggs in the box. So yeah, that's not good. It's not happening. Okay. Can you maybe explain a bit more what you mean by sophisticated behavior and what you're thinking is missing? So I'm not sure the generational choice that direct information is being passed on in some sense, you know, in a quantitative way. I don't know what's going on, right? So it could still be occurring through ecological factors. So because the first set of founder females lay a different number of eggs in that first generation, those carry-on effects may be going on. Or it's possible that something is passed, for example, epigenetically through the eggs, right? Which is then changing how every subsequent generation behaves. I don't know which of those is true at the moment. But also, I think, I also don't know whether the behaviors that we see at the end are actually optimal for each of these contexts or not. Is that making sense? Well, if we, to me, they're special, but no, I'm certain that many organisms have evolved systems of plastic behaviors to respond to environments as they, I mean, we have many examples of this, maybe not exactly in this context, but many other contexts as well. So I don't think they're special in that way.